In this video, we will discuss about this NK cells and this NKR-NK cells. NKR means that NK receptor engineered NK cells, and here the chimeric antigen receptors NK cells. So, as we have discussed in some of our videos, that in CAR could be of different generations. And most common generations that we use is co-stimulatory domain, where intercellular part has two domain. First one is CD3 and another one is the co-stimulatory domain. Both can trigger uh, both can trigger the anti-tumor response once the antigen specific tumor antigens gets activated, or the single chain fragment variable of the CARs interact with the tumor antigens. And there is another way to activate this is using this CD16, which will release ADCC response, and cytokines which could be released because of the co-stimulatory domains will then enter into the tumor cells through perforin and then trigger apoptosis. Some of the cytokines that can be regulate is granzymes, that is known for triggering apoptosis. And in the case of NK receptors, NK cells, what happens is that it will not have this single chain fragment variable. However, it will has the receptors such as NKP30, NKP46, NKGD, DNAM1, and these receptors can activated through the ligands that are present on the tumor cells. And when this is activated, it will also have this domains, that is co-stimulatory domain and the CD3 domain. So it will also once the receptors is activated or interact with this ligand with their counter ligands, then it will also trigger release of the cytokines and the anti-tumor response. And one more thing is and these receptors are generally expressed naturally in the tumor cells, and they can be overexpressed through the engineering of the NK cells. And there are some inhibitory receptors also, such as PD1 receptors, which interact with this PDL1 ligand on the tumor cells and then suppress or deactivate the NK cells. And similarly for the CD15 and this HLA-E ligand. So these things can also be modified in a way that when the PL1 ligand interact with these PD1 receptors, instead of deactivating, it will activate the tumor anti-tumor response. That can be that is shown here in the graphical form. That the regular intercellular functioning is uh, not working, or it has not been engineered. It is engineered in a way that uh, the intercellular counterparts is not present, or they can be engineered in a way that it will, instead of suppressing the NK cells, it will activate the NK cells. So it will perform instead of tumor suppression response, it will perform tumor instead of tumor suppressions, it will perform this anti-tumor response when this PDL1 ligand interact with this modified PD1 receptors on this NK cells receptor modifications. And similar modifications has also been done for CD155 receptors and HLA-E receptors. So, and whereas in the case of CAR-NK cells, the main engineer is focused on this single chain variable fragment, which is specific to the tumor associated antigens. So it will then trigger the co-stimulatory response along with the CD3 response whenever this uh, single chain fragment variables interact with the tumor antigens. And it also has the natural receptors, just like the NK receptor engineered modifications. However, in this case, there is no co-stimulatory domain, will only activate their respective pathway, such as this NKP30, NKP46, NKGD2, DNAM2. So I hope you understood the CAR-NK cells and NKR-NK cells and their difference and how they functions when they interact with the tumor cells. If you need to understand more about their uh, functions or in-depth understanding of this CAR-NK cells and NKR-NK cells, you can go through the reference that has been provided in the description. Thank you for your time.